Hi guys, Jason Matthew here again, and today we're going to be looking at amino acids and proteins, and this is part 2 of the video. So part 1 is already up on the Biochem JM channel, and in part 1 we looked at the amino acids themselves and the different chemical properties and so on, and we looked at the peptide bond. Today we're going to be focusing on the proteins themselves. So let's get straight at it. Now first, there are 4 levels of protein structure. You have primary structure, secondary structure, tertiary structure, and quaternary structure. As you go from primary to secondary to tertiary, we are looking at just one polypeptide chain. And as you go from primary to tertiary, that polypeptide chain is increased. There's increased folding in that polypeptide chain. So you start off with the primary structure, which has no folding whatsoever, to when it starts to fold and you have secondary structure, and then further folding will lead to tertiary structure. Now to get to the quaternary structure, that's where you have more than one polypeptide chain involved. Now what to remember is that all proteins will have a primary structure, all proteins will have a secondary structure. Some proteins will have a tertiary structure, and few will have a quaternary structure. So that's something to remember. It's not that all proteins will have all 4 levels of structure. It depends on the protein and its function and so on. Alright, so primary structure. The primary level of structure in a protein is the composition and linear sequence of amino acids as joined together by peptide bonds. This sequence is determined by the sequence of nucleotide bases in the gene encoding the protein. So basically the primary structure gives you the following information. It tells you what amino acids are present, and it also tells you the sequence in which these amino acids are joined together by peptide bonds. Secondary structure now. The secondary level of structure in a protein is the regular folding of regions of the polypeptide chain. The 2 most common types will be the alpha helix and the beta-pleated sheet. Let's start with the alpha helix. So let's go through this. There's a lot of information there to read, but let's just go through it step by step. In the rod-like alpha helix, the amino acids arrange themselves in a regular helical conformation. The carbonyl oxygen of each peptide bond is hydrogen bonded to the hydrogen on the amino group of the 4th amino acid, with the hydrogen bonds running nearly parallel to the axis of the helix. In an alpha helix, there are 3.6 amino acids per turn of the helix, covering a distance of 0.54 nanometers, and each amino acid residue represents an advance of 0.15 nanometers along the axis of the helix. The side chains of the amino acids are all positioned along the outside of the cylindrical helix. So a lot of information there to digest, but it's very important that you know certain things. Like for instance, we need to do a little chemistry here. When we said carbonyl, this carbonyl oxygen, do you know what group that is? Alright, so I'm hoping that you correctly said it's a C double bond O. Alright? So it's a C double bond O. It's the carbonyl oxygen. Alright? So they're saying that the oxygen in this carbonyl group, alright, is hydrogen bonded to the hydrogen on the amino group. So you all remember what an amino group is? It's NH2. So one of those hydrogens will hydrogen bond with the oxygen on the carbonyl group. Alright, and a key thing here to remember is that alpha helices are stabilized by only one type of bond, and that's hydrogen bonds. Alright, so the only type of bonding involved in alpha helices is hydrogen bonds. And these hydrogen bonds are taking place between peptide bonds. Alright? And they're very specific. It's occurring between the carbonyl oxygen of each peptide bond is hydrogen bonded to the hydrogen on the amino group of the fourth amino acid away. And these hydrogen bonds, they run parallel. Very important. Alright, next important piece of information here. For every turn in the alpha helix, alright, there's 3.6 amino acids. This comes in the multiple choice, so be very wary of that. Alright, now the hydrogen bonds themselves are in a parallel direction. The side chains, and you should know that when you looked at the first podcast, you know what side chains are. These are what we call the R groups on the amino acids. So this is what makes each amino acid different, is the presence of these R groups. And we see that the R groups are positioned perpendicular to the cylindrical helix. Alright, so what you're watching here, if you take this bar here, alright, so this bar, alright, that's an imaginary bar there, alright, and what we're saying is that the alpha helix, it looks like it's wrapping around, it's coiling around this center axis, alright. So that is basically what I want you all to get, is this coiling configuration. It's called an alpha helix. So why does the alpha helix form more readily than many other conformations? That's a good question, right? There are other conformations that is theoretically possible, but yet alpha helices are the most predominant form. And the answer to that question is that the helix forms readily due to an optimal use of internal hydrogen bonding. Therefore, within the alpha helix, every peptide bond, so remember people, I will stress this again, that for secondary structures, we're looking at hydrogen bonding between atoms in the peptide bond. Alright, so they're saying therefore within the alpha helix, every peptide bond except those close to the end of the alpha helix participates in hydrogen bonding. Each successive turn of the helix is held to adjacent turns by 3 to 4 hydrogen bonds. Alright, all the hydrogen bonds combine to give the entire helical structure considerable stability. Because if you remember from your chemistry, a hydrogen bond by itself, one hydrogen bond, is a very weak bond. But because of the sheer number of hydrogen bonds being present, it gives the helical structure considerable stability. So is it that, all amino acids are welcome in the alpha helix? Or is there some that the alpha helix would rather that it not be present? And the answer is there are two in particular that you have to look out for. Alright, now the one that we're looking at here is proline. And remember we looked at proline in the first set of lectures. And proline is special. Again, just in case you forgot, the R group for proline is this guy here. Alright, CH2, CH2, CH2. But what's special about proline is that its R group, it combines with the alpha amino group. Alright? And that gives proline some special properties. Now let's read here. In proline, the nitrogen atom is part of a rigid ring, and rotation about the nitrogen alpha carbon bond is not possible. So this nitrogen alpha carbon bond that they're talking about, it will be this bond here. Alright, so that bond there is what we call the the nitrogen carbon alpha bond. And what we're saying is that because it forms this ring-like structure there, it prevents free rotation on this bond. And what is the resulting effect of that is that the proline residue will introduce a destabilizing kink in the helix. So it destabilizes the alpha helix. Also, it cannot form the correct pattern of hydrogen bonds due to the lack of a hydrogen atom on its nitrogen atom. So, if this was a normal amino acid, this would have been NH3+. But what happens here is that because this carbon is bonded to nitrogen, one of the bonds will be between nitrogen and carbon and not between a hydrogen. So it's one short of a hydrogen as compared to the other amino acids. So it's saying that because it doesn't have, or it lacks that hydrogen, right, it cannot form the correct pattern of hydrogen bonds. Remember, the main form of stability in the alpha helix would be the hydrogen bonds. So for these reasons, proline is often found at the end of an alpha helix, where it alters the direction of the polypeptide chain and terminates the helix. So the question is now, proline, we label him as a bad boy, right? He's not welcome in the alpha helix. But the question here is, are there any other amino acids that you know of that will also destabilize the alpha helix? And yes, there is, and then one in very particular will be glycine. Alright. Now, what is the R group from glycine? And that is something you should know now, because you did the first set of lectures, proteins and amino acids, part 1, and we spoke about that. Alright, so you should remember that glycine, the R group is hydrogen, because glycine is the smallest amino acid I have there. Now here's the problem with glycine, well, in terms of stabilizing the alpha helix. Because its R group is the smallest possible atom, which is hydrogen, we say that glycine has what is called high conformational flexibility. Right? And all that, what that means is that because its R group is so small, right, it has a lot of conformational flexibility. And what in essence that does is that it destabilizes the alpha helix. So glycine is another guy who's not really welcomed in the formation of alpha helices. Alright, so let's continue. Interactions between amino acid chains can stabilize or destabilize the structure. Alright, so what we're saying is that these amino acids that are in the alpha helix, alright, they could also add to the stability, or in most times they could destabilize the structure if they're in a certain sequence. So like an example, long stretches of either negative or positive charges will repel each other. Remember, like charges will repel. So what we're saying is that if you had a long chain of, let's say, glutamic acids, you had glutamate, 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 glutamate. You had 4 glutamates in a row, one after the other. Remember the R groups about glutamate, they're negatively charged. And what we're saying is that because they're so close together, they will repel each other, and that will destabilize the alpha helix. Large bulky side chains next to each other can result in steric interference and again stop the formation of the helical structure. So again, if the R groups, they are too big and you have these bulky R groups close to each other, you wouldn't get that proper coil where you have 3.6 amino acids per turn. Reason being is that because if the R groups are too bulky, they can't fit close enough and they can't give you that proper turn. So that will also destabilize the alpha helix. Last but not least, the twist of an alpha helix ensures that critical interactions occur between an amino acid side chain and the side chain 3 and sometimes 4 times away on either side of it. Positively charged amino acids are often found 3 residues away from negatively charged amino acids, permitting the formation of an ion pair. Alright, 2 aromatic amino acid residues are often similarly spaced, resulting in a hydrophobic interaction. So these interactions and stuff, they further stabilize the alpha helix. Right, because remember, like the 2 amino acids that will come close together is 1 amino acid and then one that is about 3 to 4 residues away. They're going to come close together in that when you make that coil. Alright, that turn. within one turn, the amino acid, let's call it 1, and then the one that is 3 to 4 residues away is going to be close to it. So you could have some interactions that would further stabilize the alpha helix. So you could have the formation of an ion pair if the R groups, alright, if one R group is negatively charged and the other R group is positively charged. Or sometimes if you have two aromatic amino acid residues coming close together, right, it could result in a hydrophobic interaction. All these things will further stabilize the alpha helix. The final factor affecting the stability of an alpha helix is the identity of the amino acid towards the ends of the helix. The small electric dipoles of each polypeptide bond are connected to each other through hydrogen bonding, resulting in a net dipole along the helix that increases with helix length. The 4 amino acids at the end of the helix do not fully hydrogen bond. Thus, negatively charged residues are found at near the amino terminal, where they interact with the positive charge of the helix, stabilizing the structure. Positively charged residues are found at the carboxyl end, where they stabilize the negative end of the helix's dipole. So let's look at the diagram here. What we're saying is that, remember, the main stability or stabilizing force in an alpha helix is all these hydrogen bonding between the peptide bonds. Now each hydrogen bond will have a small dipole being set up, and because the hydrogen bonds are in a parallel arrangement, these dipoles will line up to form a macro dipole. And when the macro dipole is formed, you will see that the N-terminal end will be slightly positive and the C-terminal end will be slightly negative. So to stabilize that macro dipole, what you see happening is that amino acids that are close to the N-terminal end will be negatively charged so as to stabilize this positive charge. And likewise, amino acids close to the C-terminal end will be positively charged so that it can stabilize this slightly negative charge. Alright, so you see a further picture here, and you can give this a read. Alright, it's basically the same thing I've been saying before. So let's summarize the 5 factors affecting alpha helices. The occurrence of proline and glycine residues, the electrostatic repulsion or attraction between successive amino acid residues with charged R groups, the bulkiness, or in other words, the steric hindrance of adjacent R groups, the interactions between R groups spaced 3 or 4 residues apart, and lastly, the interaction between amino acid residues at the ends of the helical segments and the electric dipole inherent to the helix. Now we go to the secondary structure available, which is the beta-pleated sheet. Now in the beta-pleated sheet, hydrogen bonds form between the peptide bonds either in different polypeptide chains or in different sections of the same polypeptide chain. The planarity of the peptide bond forces the polypeptide to be pleated, with the side chains of the amino acids protruding above and below the sheet. Now your beta-pleated sheets could be either parallel or perpendicular. That depends on your C-terminal, N-terminal end orientation. So here we have like 3 polypeptide chains and we have the hydrogen bonds taking part in them. Now if you look at the side view, the purple will represent the R group, so you see that they are above, below, above, below, so they alternate. Alright, now this red here should represent the C-terminal end, while on this side it will be the N-terminal end. Alright, so you see, like, if you can watch carefully, you see a C double bond O, C double bond O, C double bond, and then you see these blue here will represent the amino groups. Alright, now if you watch carefully, on either side, all the C-terminal ends, they line up. All the N-terminal ends, they line up. So we say that when you have the same end, on each of the polypeptide chains, like you have all the N terminal lining up or the C terminal lining up, we call that parallel beta-pleated sheets. And anti-parallel beta-pleated sheets, I think before I slipped up and I said perpendicular, that's wrong. So just in case if I said that before, that's wrong. You have parallel sheets, which is the ones we looked at before, and now we're going to be looking at anti-parallel beta-pleated sheets. And the anti-parallel beta-pleated sheets, the C terminal and N terminal ends, they alternate. So if you watch here, you have a C terminal end. But for this polypeptide chain, it's an N terminal end. And then for this one, it's a C terminal end. So you should guess what would be next if we continue the trend. Here would have been an N terminal end, right? And similarly on this side, it would be N terminal, C terminal, N terminal. So they're alternating, and we say that's anti-parallel, all right? Now we go to the good stuff. Tertiary structure. The tertiary structure refers to the spatial arrangement of amino acids that are far apart in a linear sequence, as well as those residues that are adjacent. In water-soluble globular proteins such as myoglobin, the main driving force behind the folding of the polypeptide chain is the energetic requirement to bury the non-polar amino acids in the hydrophobic interior away from the surrounding aqueous hydrophilic medium. Right, so that's very, very important of what we're saying there, right? What we're saying is that the main driving force for the three-dimensional structure or the tertiary structure in a globular protein is what we call the hydrophobic effect. Now let's just recap a little bit. When we looked at amino acids, we saw that some R groups could be polar, which we will call hydrophilic, and some R groups will be non-polar, which is hydrophobic. They don't like water. So if you put them in a hydrophilic medium, like in a water medium, the non-polar amino acids wouldn't like that medium. So it wants to bury itself down into the middle, all right? So we say that the protein folds in a way so as to only expose the polar, or in other words, the hydrophilic amino acids to the water medium, while the non-polar hydrophobic amino acids will bury themselves in the center, all right? And that driving force to do that to get away from the hydrophilic environment, all right? We call that the hydrophobic effect. All right, so polypeptide chain folds spontaneously so that the majority of its hydrophobic side chains are buried in the interior and the majority of its polar charged side chains are on the surface. We call that the hydrophobic effect. Once folded, the three-dimensional biologically active native conformation of the protein is maintained not only by hydrophobic interactions but also by electrostatic forces, hydrogen bonding, and if present, the covalent disulphide bonds. So just to make a point here, the most important driving force for the three-dimensional structure of a protein is the hydrophobic effect, and the hydrophobic interactions are the most important bond that stabilizes and gives the protein its three-dimensional structure. All right, so that is the most important one, but it is not the only force that keeps the protein in its three-dimensional shape. There are other forces of attraction involved. For instance, electrostatic forces of attraction, hydrogen bonding, and if present, covalent disulphide bonds. Now, don't think of a hydrophobic bond as being very, very strong. That's not true. In fact, the covalent disulphide bonds will be the strongest out of the lot here. But covalent disulphide bonds do not contribute significantly to the stability of the proteins. Although they are the strongest bond present, they are not frequently found in a protein. All right? But you will always have hydrophobic interactions. And while one hydrophobic interaction, if you want to count it like that, while one of them might be small, it might be weak, combined, because the sheer amount of hydrophobic interactions taking place, it plays a very significant contribution in the stability of the three-dimensional shape of the protein. So here we look at the different bonds involved. All right, we have a hydrogen bond. And remember, if you remember chemistry, a hydrogen bond is formed from, as the name suggests, from a hydrogen atom as well as a very electronegative atom, such as oxygen and nitrogen. Also fluorine if you go in the chemistry way, but for most biological systems, it'll be oxygen and nitrogen involved. Now, hydrophobic interactions where you have two nonpolar groups interacting with each other. So if you watch here, both of them are nonpolar. The ionic bond, or what we call the salt bridge, is between oppositely charged R groups. And then the disulphide bridge is something that we looked at in the first set of lecture. It's very important that you know what two amino acids come together to form a disulphide bridge. I will leave that for the first set of lectures, so please go back to proteins and amino acids part 1, and just in case you can't remember it. Now, let's just backtrack a little. When we looked at secondary structures, we were saying that it was mainly formed due to the hydrogen bonding taking place between atoms in the peptide bond. Now, for the tertiary structure, we are looking at interactions among the R groups of these different amino acid residues. So it's these interactions between the R groups when it comes to tertiary structure. Alright, so remember, hydrogen bonding in the peptide bonds is what's contributing to these secondary structures, and now for tertiary structures, we're looking only at the interaction between R groups of different amino acid residues. And if you watch here, alright, if you look here, this is a very good example here. Look at what's happening with tertiary structure. You have an amino acid residue here that is interacting with an amino acid residue over here. Now, if you look at the primary structure, these two amino acid residues are extremely far apart. But yet, because of the folding and so on, because of the three-dimensional shape, you have two amino acids that are relatively far apart interacting with each other. Very important. So let's look at these forces individually now. And I think I did a good job before. You should already have a very good idea about these forces. But let's just reinforce that information now. The hydrophobic effect is a name given to those forces that cause nonpolar molecules to minimize their contact with water. Right? So by burying those nonpolar hydrophobic molecules into the center there where they are away from water, what we do is that we increase the entropy of the system. Alright, and let's remember, when you increase entropy, there's increased stability, so it is energetically favored. Alright, and the hydrophobic effect is the major driving force for the three-dimensional folding of proteins. Always remember that. This is clearly seen with amphipathic molecules, and you should know what amphipathic is. I'm not going to give it away, so if you don't know what amphipathic is, please go and look it up on the net. If you Google it or you Wikipedia it, you're going to get an answer right away. So I'm not going to give away that, so check it out. Alright, such as lipids and detergents, which form micelles in aqueous solutions. Now, if you're not too sure about micelles, don't panic. We haven't done lipids as yet. When we do lipids, we will briefly look at the formation of micelles, so don't panic too much about that. Proteins too find a conformation in which their nonpolar side chains are largely out of contact with the aqueous solvent, and thus hydrophobic forces are an important determinant of protein structure, folding, and stability. Alright, so hydrogen bonding now. In biological systems, the donor group is an oxygen or nitrogen atom that has a covalently attached hydrogen atom, and the acceptor is either oxygen or nitrogen. Hydrogen bonds are normally in the range of 0.27 to 0.31 nanometers and are highly directional. That is, the donor hydrogen and acceptor atoms are collinear. Very important. Hydrogen bonds are stronger than van der Waals forces, but much weaker than covalent bond. So, quick question. What is an example of a covalent bond in tertiary structure, proteins? Alright, so you should quickly say an example will be disulphide bonds, and you will be absolutely correct. So I hope you said disulphide bonds. Hydrogen bonds not only play an important role in protein structure, but also in the structure of other biological macromolecules, such as the DNA double helix and lipid bilayers. In addition, hydrogen bonds are critical to both the properties of water and to its role as a biochemical solvent. Electrostatic forces. And we saw that where you have opposite charges, they attract. Alright, so for example, the ammonium group of lysine will be positively charged, and the carboxyl group of aspartate will be negative charge. Remember, these, when I say amino group and carboxyl group, I'm talking about the R group on these individual amino acids, right? So the R group of lysine will contain an amino group, and the R group of aspartate will contain a carboxyl group. The amino group and the carboxyl group that we are talking about is not the alpha amino group or the alpha carboxyl group, right? So that's to make sure. I'm going to spoil you a little bit and let's do a little quick revision, although it's something that you know. But the general formula for an amino acid is CR, alright, let's say NH3 plus COO minus and SAH. Alright, this is the alpha carboxyl group, this is the alpha amino group. When we say that it's opposite charge, for example, the amino group of lysine, we are not talking about this amino group. We are talking about there's an amino group in the R group, right, of lysine. Similarly, for the aspartate, within this R group, there's a COO minus, all right? And what we're saying is that the charge, the opposite attraction there, will cause a salt bridge. So that's a form of attraction and a form of between the R groups. So always remember, tertiary structure is all about force of attraction between the R groups. Now you already remember the disulphide bonds. We looked at it in the first set of lectures. So I won't go into it too much, but just remember. So in case you need any more information, go back to the first set of lectures and look back at them. We spoke at more detail there. But just remember, two cysteine amino acid residues will come together to form cysteine. And when cysteine goes to cysteine, that's the oxidation reaction, and a disulfide bridge is formed. Now you can easily reverse that process, and to reverse that process you need to do a reduction where each sulfur will gain a hydrogen, so you form back these two cysteine molecules. So that's something to check out. Now sometimes, just changing one amino acid in a protein could prove to be very dangerous. So let's look at here. Sickle cell anemia. A small change in the sequence of the primary structure can have a significant impact on protein structure. So in sickle cell anemia, glutamate is replaced by a valine in the amino acid sequence. Now what I want you all to do here is go back to your notes, because most likely you can't remember it often. That's fine. Go back to your textbook, go back to the notes, go back to the podcast, and look at what is the structure for glutamate, and look at what's the structure for valine. And my other question is based on the information that you have just gathered, alright, and what you know about the hydrophobic effect and so on. What you need to ask yourself, would glutamate have been on the surface of the protein or within the center? And why it is that glutamate will be outside and not on the inside? And then the next question is now, well, what is the significance of valine? Will valine be able to do the same thing that glutamate was able to do? And that should give you some answers to why sickle cell hemoglobin will be problematic, right? Last but not least, we're looking at the quaternary structure and proteins, and here's the pointer. From primary, secondary, and tertiary, we'll just deal with one polypeptide chain. For the quaternary structure, we're looking at least 2 polypeptide chains. It could be much more. Alright, and the classic example of quaternary structure would be hemoglobin. Alright, the level of structure refers to the spatial arrangement of the polypeptide subunits and the nature of the interactions between them. Alright, and the same forces of attraction that stabilize and cause the tertiary structure. So we're looking at hydrophobic interactions, hydrogen bonding, electrostatic forces, disulphide bonds. The same set of forces that were involved in tertiary structure will be the same set of forces that are involved in keeping the quaternary structure together. So why quaternary structure? Why it is that we didn't just leave these separate polypeptide chain just to exist on their own, because they can, Why it is that, what is the benefit of having an association between these different polypeptide chains? Now, we say that the protein is what we call an oligomeric protein, meaning that it's made up of more than one polypeptide chain. So let's read and see what's happening here. Oligomers are usually more stable than their dissociated subunits. So we see here that the quaternary structure offers more stability, suggesting that the quaternary structure prolongs the life of a protein in vivo. The active sites of some oligomeric enzymes are formed by residues from adjacent polypeptide chains. The three-dimensional structure of many oligomeric proteins change when the proteins bind to ligands. Both the tertiary and quaternary structures may be altered. Such changes are key elements in the regulation of biological activity of certain oligomeric proteins. Remember, oligomeric means that the protein is made up of more than one polypeptide chain. Different proteins can share the same subunits. Since many subunits have a defined function, evolution has favored selection for different combinations of subunits to carry out related functions. This is more efficient than selection for an entirely new monomeric protein that duplicated part of the function. And last but not least, another very useful role of quaternary structure will be a phenomenon called substrate channeling. So please do a little reading and see what substrate channeling is all about. So let's just talk a little bit about the denaturing of proteins. And we have all seen it with our eyes. We probably didn't really think at the moment that this was a protein being denatured, but I'm sure all of us have observed the denaturation of protein to some level. So let's read. The natural or native structures of proteins may be altered and their biological activity changed or destroyed by treatment that does not disrupt the primary structure. Right? So let's remember that when we're talking about denaturation, we are referring to the loss of folding. Right? We're referring to the loss of tertiary structure and to some extent secondary structure. But always, the primary structure always remains intact. Alright, so there's never any hydrolysis of peptide bonds taking place when the protein is being denatured. That's something different. Alright, so the primary structure remains intact. That is very important. Following denaturation, some proteins will return to their native structures under proper conditions, but extreme conditions such as strong heating usually causes irreversible change. So what we're saying here is that whatever is denaturing the protein, in other words, the protein is unfolding. If you were to remove that denaturant, most likely the protein will be able to refold back into its native structure and it will be able to work again. So some common denaturation agents. We have heat, very common, we all are familiar with that. And remember, in denaturation, or when something is being denatured, it means that the protein is starting to unfold. Now, for the protein to start to unfold, it means that the bonds that was keeping the tertiary structure intact are being broken, right? So you'll see that these different denaturing agents is precisely doing that. They are breaking the bonds that keep the tertiary structure together. Alright, so you see for like heat, hydrogen bonds are broken. by increasing the translational and vibrational energy of the hydrogen bonds. Alright? And you have other denaturants. You have UV radiation, you have strong acids or bases, you have urea, you have organic solvents, or just simple agitation, meaning shaking it up, could cause denaturation. So please have a read of this, and then we can move on. So make sure you go through each one in the table here. They're basically self-explanatory, so I'm not going to go through each one. I want you to pause the video and go through each one for yourselves. Right, so heat denaturation. Most proteins can be denatured by heat, which affects the weak interactions in a protein, primarily hydrogen bonds in a complex manner. So basically, for heat denaturation, the main bond that is being broken here will be hydrogen bonding. If the temperature is increased slowly, a protein's conformation generally remains intact. So if we are increasing temperature slowly, we will not see denaturation happening gradually. What really happens is that most of the times is that there's suddenly an abrupt loss of structure and function, and it occurs over a narrow temperature range. So what we say is that as you start to increase the temperature, the protein is able to withstand that increase in temperature. But it has a threshold, and once you pass that threshold, then there's a certain abrupt loss of function. Alright, so the proteins they denature over a narrow temperature range. Right, and we will see this when we look at the effect of temperature on enzymes. Remember, enzymes are proteins, and you'll see this abruptness once more. You'll see the curve and everything like that. The abruptness of the change suggests that unfolding is a cooperative process, meaning that, that as soon as it's like the protein is able to withstand the increase in heat as it gradually increases. But it will reach a point that as soon as one part of the protein gets destabilized, the whole protein just quickly becomes destabilized as well. So it's like as soon as you pass that threshold, boom, as one part starts to crumble, all the rest will start to unfold as well. Right, so you see in the top part here, you have a protein that has this three-dimensional shape. It's tightly folded, and then when you apply heat, the hydrogen bonds, they are disrupted, and there's unfolding taking place, alright? But the primary structure will remain intact, which is the sequence of the amino acids. So solubility is drastically decreased as in heating egg whites, where the albumins unfold and coagulate. And as I said before, we're going to be looking at the effect of temperature on enzymes, because enzymes are proteins as well, and you'll see that they could become denatured as well. So now we have chemical denaturation. So we looked at some physical forms. Let's look at what chemicals we could use in the lab to denature a protein, because sometimes that's our job. We need to denature the protein to study them. So proteins can also be denatured by two types of chemicals. You have the charotropic agents and you have detergents. Some examples of the charotropes will be thiocyanate, perchlorate, alright? So those are some of the major examples of charotropes. Thiocyanate, perchlorate, and I would just like to point out as well, another useful guy will be urea, alright? And what charotropes do? They enhance the solubility of non-polar compounds in water by disordering the water molecules, right? High concentration of molecules such as urea denature proteins by allowing water molecules to solvate non-polar groups in the interior of the protein. And this is the real take-home message here that I want you to always remember. The water molecules disrupt the hydrophobic interactions that normally stabilize the native conformation. So for example, in the exam, if I ask you what is the role of urea in protein chemistry, what you should say right away is that urea is a charotrope, and a charotrope will disrupt the hydrophobic interaction. Therefore, the protein will lose its tertiary structure and it will lose its activity as well, if it's an enzyme, that is. Last but not least, the hydrophobic tails of detergents such as sodium dodecyl sulfate, or widely known as SDS. They will also denature proteins, and they do the same way in a similar way to the chirotropes, in that the detergents will disrupt the hydrophobic interactions. And remember, what if you remember from your previous slide, the major driving force for proteins, for the tertiary structure of proteins, will be hydrophobic interactions. So once you disrupt these hydrophobic interactions, the main driving force in keeping the protein in its three-dimensional structure, what's going to happen is that once you disrupt the hydrophobic interaction, the protein is going to start to unfold. So then we have this brilliant guy, Finson, and he did a very interesting experiment. Finson was among the first to demonstrate that denatured proteins can refold spontaneously to their native conformation. This indicates that the information required for the native 3D conformation is contained within the amino acid sequence of the protein. In other words, and please make sure you get this, because this is the take-home message from the Finson experiment. This is what he was able to conclude. So he concluded that the primary structure determines the tertiary structure. In other words, the primary structure, that sequence of amino acid, it contains all the information that is necessary for the protein to fold into its three-dimensional structure, which is the tertiary structure. So here is his experiment, and if you could follow it, that would be great. He used an enzyme called ribonuclease, and he was lucky in that that enzyme was a small, simple enzyme to study. So you have the protein. If you look on this side, you have the protein in its three-dimensional active structure. So the enzyme is active there. What he did is that he added, well, in his experiment, he added guanidinium hydrogen chloride plus mercaptoethanol, or in some books you see it's called beta-mercaptoethanol. Alright, same thing. Alright, and what this did was that it denatured the protein. Now you have two chemicals here, and these two chemicals are doing two different things. The guanidinium hydrogen chloride is an example of a chirotop. And what the chirotop would do, it would disrupt the hydrophobic interactions. Now another chemical that could have been used here would have been urea. Urea is also a chirotop, and it will do the same function. It will serve the same function as guanidinium hydrogen chloride did. It will disrupt the hydrophobic interactions. But the thing is that when you disrupt the hydrophobic interaction, most of the protein will unfold. But if the protein had disulphide bonds, then they'll have, wherever the disulphide bond is, it is still going to keep some of the protein in that folded form. So that's why you add the second chemical, which is mercaptoethanol. And remember how to break a disulphide bond? You have to reduce it. Alright, good job. So what beta-mercaptoethanol does is that it reduces the disulphide bond. So in other words, it breaks the hydrogen bond. The disulphide bond, sorry. But do not say the word break disulphide bond in the exam. Eh? Be more sophisticated than that. Say it reduces. So beta-mercaptoethanol is a reducing agent, and it would reduce the disulphide bond in a cysteine molecule into two cysteine amino acids. And that's when you're showing off some swagger there, if you could say that in one shot. Alright? Now here's the thing. So when he added, in his experiment, he added guanidinium hydrogen chloride and beta-mercaptoethanol, the protein unfolded. And when the protein unfolds, it loses its catalytic activity. Remember, it was an enzyme he was working with ribonuclease, so he could have measured and see if the resulting solution had any enzymatic activity, and he saw that there was no activity, so he concluded that the protein unfolded. But here's the thing, the next step of the experiment now, he used what is called dialysis. And simply put, what dialysis is that you're going to put in a tube and you're going to put this solution here. So this solution here would have contained the unfolded protein, and it would contain guanidinium hydrogen chloride and beta-mercaptoethanol. Now the dialysis tubing sometimes look like a pellicle pack, so you put the solution in that. Now the dialysis tubing has very small pores in them, and these pores allow small molecules to pass through. So what happens is that when you put this dialysis tubing in a beaker full with buffer, is that the guanidinium hydrogen chloride and the beta mercaptan, they are small molecules, so they're able to pass through the pores in the dialysis tubing. The ribonuclease protein, or the unfolded protein, is too big to pass through the pores, so the protein remains in the dialysis tubing. So you have the beta mercaptan and the guanidinium hydrogen chloride, they are going through the pores and they're coming out of the dialysis tubing, but the proteins, they are too big to pass through the pores, so they remain inside the dialysis tubing. And basically, the guanidinium hydrogen chloride as well as the beta mercaptan ethanol is moving out of the dialysis tubing because of diffusion. So you always want to maintain that diffusion gradient. So what is recommended is that you change the buffer solution every now and then. Alright, now if you continue that process, you're going to have a time when now all the guanidinium hydrogen chloride and all the beta mercaptan ethanol has been removed. And as soon as you remove all the denaturation agents, what will happen is that the unfolded protein, remember the primary structure was never interfered with, the primary structure remained intact. And the primary structure contains all the information for the protein to fold into this tertiary structure. So what Affinson observed is that once he removed the guanidinium hydrogen chloride and the beta mercaptan ethanol, once he removed that, it was no longer present to denature the protein. As soon as you remove it from dialysis, the protein was able to refold into its proper three-dimensional shape. And once it formed its proper three-dimensional shape, the active site was reformed and it made the protein active once more. So that's, in a nutshell, what a Finsen experiment is all about. Alright, so you can look at this here. This is, I think it was taken from the ledger. Don't quote me, I might be wrong. But you see here again, the protein is folded in its three-dimensional shape. And when it has this proper three-dimensional shape, the active site will be in its proper shape for proper enzyme activity. When you add the guanidinium hydrogen chloride in this example, or well, in this example they use urea, alright? So great, urea will act as a karyotrope, and like any other karyotrope is going to disrupt the hydrophobic interaction. Therefore, most of the protein will start to unfold, because that's the hydrophobic interaction is the main driving force for the three-dimensional folding of a protein. So once you remove that, the protein starts to unfold. But there will still be some disulphide bonds that may be present that will still try to keep the structure together. So that's why you're going to use a second chemical called beta mercaptan ethanol, which will reduce these disulphide bonds. Alright, so in other words, you're going to be breaking the disulphide bonds in layman terms, that is. And once you do that and the protein totally unfolds, it means that it loses the active site. Alright, the enzyme loses its active site. Therefore, the enzyme becomes inactive. Alright guys, so I really hope that you all stayed to the end with this. Alright, now as you can see, this could be the biochemistry guy, and sometimes this is all to you and you think it's the odds are way against you. The biochemistry is looking at this so much. But what I will continuously urge you to do is to never give up. Just keep at it. Keep studying every day. Try to take a little bit of notes, and always, keep writing, keep doing questions, practice. That's how you're going to make perfect. And just keep at it. Never give up. Right? And as usual, if you like what you heard, please show your love by hitting the like button. Also, please subscribe to our channel. Alright? There are a lot of other informative videos, so please go give it a check. I would love to hear what you all think about these videos, so your comments are greatly appreciated by myself. Alright, you can also join me on Facebook. I have all my students there. I would love to form an international network with all students all over the world, just hearing what their experiences in learning biochemistry and giving us tips and clues on how you all do biochemistry away, and let us improve how we teach down here, So if you have any information like syllabus and slides and information that you use that you think will be helpful for us, please send us your info at to jasonmatthew2011@gmail.com. Alright, let's share this information. You saw what we do here. Please send some information about how you do things so we can get a whole different look at the topics, and improve our teaching. So I look forward to that. And as usual, people, thank you for listening to this video. I really hope it helps. And as I said before, don't give up. Keep at it, and we'll talk very soon. So take care, guys.